I was with my sister and her family at VINS, and she said, how come they don't know what sex these birds are? I said, oh yeah, because not all birds are obviously different.
Some you can easily tell the difference between a male and a female. So some have wildly different sizes or feather patterns that can tell you at a first glance whether or not you're looking at a guy or girl. But some are a little more subtle and not sure, and some are pretty much non-existent.
Emily, the professor at Landmark College, had reached out to the team here at VINS and realized that some of our birds here at VINS did not have their sex known. And knowing that she was a professor in Principles of Biology course at Landmark College, she was able to kind of come up with an idea that her students in that course would be able to run some tests.
Kim Coleman is teaching another section of this class, and I proposed it to her as well. And we all got excited about the idea of giving our students, a real-world application for doing a lab, a couple of labs in an introductory biology class.
So we used gel electrophoresis back at the college to run the DNA.
If they're ever going to work with DNA, they're going to need to know how to do that. And so any biotech field, and a lot of biology fields, even people that think they're going into environmental science might end up doing like environmental DNA because it's a really useful tool for monitoring biology.
First, start out by pipetting on DNA, mixing it with fluids, buffers, so it doesn't mess it up. When you use the pipette, you suck up the liquids, and then you carefully put it in each capsules. We have little capsules. Then we put it in the PCR machine. And then later on, we test it with the UV lights.
They're going to have to understand concepts like DNA extraction, PCR. It's ubiquitous. It's everywhere. Everybody does it. It's the basis for a lot of biotechnology.
We all kind of drew a bird randomly. Everyone was very excited. We all got very attached to our birds despite not having met them.
Some students had positive controls, and they were very excited that we got the correct result. I didn't tell them they had positive controls until after we got the data back. So it was satisfying to see them say, oh, hey, we got the kind of, we got the sex chromosomes we expected, and it matches with reality.
So what we did is we came over here, and after these people were very nice, they generally had some feathers, we ran the DNA through that, and then we actually got to meet the birds and finally tell them for sure whether they had guys or girls. My bird we met earlier, it's the red-tailed hawk. My bird's name was Bloomfield. And it's a very big bird, even for a female, because the sexual dimorphism that you can find in that species is females being larger than males. But even within that, Bloomfield is quite a large bird.
The students who have visited today have been awesome and nothing but excited to experience VINS and kind of ask questions and see in real life what their research within the classroom has done in the community. It has some real world effects, which is a really great way for these students to know that their work in class has true benefit.
For the most part, really fun. There was, some challenges in the lab reports we did, but that's expected. And today was really fun, walking through the birds and the trail. I loved that.
They've been enthusiastic about the process. They see the point. I believe they see the point.
It's no longer just a test sample, it's a living, breathing bird that's right in front of you. So I just think it was really cool.